Hello, everyone. I would like, first of all, to thank the organizer of this conference for giving me the opportunity to address such distinguished audience. Let me introduce myself. I am Alanoud, the Assistant Professor of Plant Ecology in the Botany Program at King Saud University in Riyadh, Saudi Arabia. In this talk, I would like to concentrate on transcriptomic change in wheat under water stress during 3 life stages. My talk is particularly relevant to those of you who are studying the regulation of physiological pathway under drought stress. In this presentation is structured as follows: the introduction, then the workflow and results, conclusion, and take-home message. The Kingdom of Saudi Arabia is among the top countries in terms of agriculture in desert area. The country uses highly developed agriculture technologies to grow different crops under challenging environmental conditions. The global climate change and the consequent increase in temperature and drought, especially in the arid and semi-arid regions, made the situation even more challenging. This work aimed to determine the physiological and molecular mechanism underpinning drought tolerance in wheat using 2 local Saudi wheat cultivars. Understanding the physiological response and gene regulation under water stress could contribute to improving wheat cultivation in Saudi Arabia. Here we explored the expression patterns of transcript and two cultivars under lab conditions. During 3 growth stage, vegetative, flowering, and green filling stage, by conducting comparative analysis of different in the transcriptomic under both control, which is well watered conditions, and water stress created by 15% pH 6000. At 3 different levels of study: morphological, physiological, molecular. The first experiment was to assess drought tolerance in the 2 wheat cultivar grown in different regions of Saudi Arabia by comparing their growth and yield under well water conditions and water stress conditions. Shoot weight and length, root weight and length, root shoot ratio, RWC, proline content, soluble sugar content, and protein content, in addition to yield, were used as a selection criteria for drought resistance. The results confirmed that Najran is drought resistant and Rafah is drought sensitive. In the second experiment, RNA sequencing was performed in leaf samples harvested from water control and treatment plants of both cultivars at the 3 growth stages. Okay. To obtain maximum information, selecting a suitable workflow and analysis tools for the massive amount of data generated by next-generation sequencing is crucial. In this research, the experiment designed to find differential expression genes based on RNA-seq is alignment or mapping of short sequencing reads, FASTQ file, against wheat reference genomes from the National Center for Biotechnology Information. The library preparation, we used here a depletion methods designed to remove ribosomal RNA, leaving all messenger RNA for the sequencing. We used several software packages such as Salmon tool for reading quantification and mapping. And also we used with our package DC2, the differential expression normalized, gene annotation, and Go terms were generated. Metabolic pathway generated by KEGG analysis. The RNA-seq results were confirmed by a real-time qPCR by selecting 4 genes related to the highly regulated metabolic pathways. This experiment yielded 24.2 gigabyte of sequencing data. At least 40 million reads of 85 per piece each were obtained per sample. Although genes from different pathways change expressions under water stress, the increase in expression of genes associated with photosynthesis, amino acid metabolism, and secondary metabolism, such as phenol propionate. Okay. Here, morphological characters have shown apparently, the differences of water stress effect in both cultivar and plant growth rule. Okay. The differential regulation of the transcriptomic increase between Najran, which is a drought resistant, and Rafa, which is a Rafa cultivar drought sensitive, under peak treatment with the leaf age. Okay. Most of the differential expression genes regulated under water stress were found to be involved in metabolic pathways and the biosynthesis of secondary metabolism in both cultivar at all growth stage under study. Okay, here. This relates to the fact that water stress induced plant cell dehydration, passive water loss, then accumulated the secondary metabolic substance called active osmotic adjustment to increase the cell water balance under drought stress. These figures showing that most regulated biological process is phenyl propionate biosynthesis. So here, from KEGMAP, we found that regulated enzymes are phenylalanine aminolase in the phenyl propionate biosynthesis. Okay, we are in conclusion. To sum up, we examined the effect of 15% PX6000 on 2 wheat cultivars collected from Najran and Rafa during 3 growth stage. Our results show that genes related to water stress significantly regulated in both cultivar under study. The Najran cultivar at the vegetative and green field stage has significantly high gene expression related to the phenol propionate biosynthesis and the lignin catabolic process than the Rafa cultivar. This study confirmed that Rafa wheat cultivar was more sensitive than the Najran wheat cultivars to 15% PX6000. This showed that wheat's adaptation capacity to water stress depends basically on the cultivar genotype and development stage according to the differential gene expression profile. Comparison analysis shows that Saudi cultivar under water stress have a high expression of gene known to be upregulated under drought stress. Thus concluded that Najran cultivar is resistant to drought compared with the Rafa cultivar and could be promising sources to improve wheat drought resistance in a breeding program. We are here. Okay, there's some take-home message. Based on my research and review literature, the vulnerability of wheat plants to water stress is higher at the flowering stage compared to the vegetative and grain filling stage. Attempts to improve drought tolerance in wheat should be targeted to these growth stages. The phenol propionate metabolic pathways plays a key role in resistance to water stress in wheat and might be a target for improving drought resistance in this crop. Okay. Thank you. Well, this brings me to the end of my presentation. Thank you for your listening. If you have any questions, just send them to me, just send them to my email here and I will be happy to answer them. Thank you. Well, this brings me to the end of my presentation. If you have any question, just send them to my email here, and I will be happy to answer them. Thank you for your listening.